Hello, everyone. My name is Aamer Mahmood. I'm a senior scientist at the Berkeley Nanotechnology Center at Purdue University. And I'll be presenting a work which has been going on for a few years now with collaborators at the Indiana University School of Medicine. It's titled Wireless Measurement of Sympathetic Nervous Activity Using Planar Nanoelectrode Arrays. My presentation today, I will start with a brief introduction to the nervous system, then make a statement on why we are doing this work. We'll have a brief discussion on how we make our devices in the fabrication process flow. We'll talk about the measurement of nervous activity and a few slides towards the end stating the summary as well as what we plan to do in future and acknowledgements. So introduction to the nervous system. The nervous system is an organ system in humans and animals. It receives stimuli and produces appropriate responses. It is composed of intercommunicating cells called neurons. The figure you see on the screen here, coming from the left towards the right, we show a single neuron. This is a motor neuron, and it's a motor neuron because it terminates in muscle fiber and affects motion over there. So coming from left to right, we have, first of all, the dendrites, which are meant for communication with the next neuron upstream in the neural path. Information is transmitted in this direction. We have the cell body, which contains the nucleus. Then we have the axon. And finally, we have a termination in muscle fiber. This is a motor neuron because it affects motion. Another version could be a sensory neuron, which senses, in which information would flow in the opposite direction, upstream towards perhaps the central nervous system. Let's go briefly over the nervous system. It's composed of two parts. There's the central nervous system, which consists of the brain and the spinal cord. And it is responsible for complex functions like speech, vision, balance, and thought processes. And then there's the peripheral nervous system. Now, everything but the brain and the spinal cord make up the peripheral nervous system, and it is responsible for communication between the central nervous system and the rest of the body. The peripheral nervous system, in turn, is made up of the somatic nervous system as well as the autonomic nervous system. The somatic nervous system is responsible for voluntary control functions. I turn the laser on, I turn it off. This is something that I do voluntarily. So this is done by the somatic nervous system. The autonomic nervous system, on the other hand, controls involuntary body functions, like heart rate, digestion, respiration, etc. So even when a person is not conscious or asleep, these functions do carry on, and they are the responsibility of the autonomic nervous system. The autonomic nervous system, in turn, is composed of the sympathetic nervous system and the parasympathetic nervous system. The sympathetic nervous system is responsible for adjusting the body in response to stressful situations like cold, exercise, fear, or trauma. And it generally has an effect to increase the activity of the target organs, like increase in the heartbeat in case of fear, for example. The parasympathetic nervous system, on the other hand, has a calming effect, maintains essential body functions like digestion and excretion, etc. Now, for electrical engineers, I like to make the analogy of a temperature controller where you have a heating portion and a cooling portion, and the job of the controller is to get inputs from both of them to kind of maintain a temperature. Likewise, a healthy nervous system maintains homeostasis by balancing inputs from both of these branches of the autonomic nervous system during activities, different kinds of activities ranging from relaxation to walking, and exercise, etc. So just to recap, we went through a few slides about the nervous system, made up of the central and the peripheral nervous systems. The peripheral nervous system, in turn, made up of the somatic and the autonomic nervous system, and the autonomic nervous system made of the sympathetic and parasympathetic nervous systems. So what's the objective of this work? We are, especially my doctor friends, they are interested in exploring a biophysical relationship between sympathetic nerve activity and life-threatening cardiovascular diseases. So we want to basically understand the language with which the nervous system communicates with the organs, which we call the sympathetic tone, and we want to improve our understanding of the sympathetic tone. Why do we want to do this? Because the hypothesis is that if we can understand that language, we would be placed in a position to make a diagnosis earlier, before a heart attack, for example, begin treatment, and thus save lives. How do we propose to do that? Well, one of the problems with measurement of nervous activity, which is not new, it's been there for many decades now, is the poor quality of the signal-to-noise ratio. That's been a problem. So we plan to attack that. The first step we felt was to improve the signal-to-noise ratio, which is given by this expression, very well known to everyone here, I believe. And how did we plan on doing that? We said, let's attack the limiting noise, which is the Johnson noise. And it's given by this expression that you see. It's directly proportional to the square root of the resistance. And we said the dominant resistance that's going to be part of our measurement circuit is going to be the electrode-electrolyte interface, where our electrode actually sits on a nerve. So we plan to increase the surface area of our electrode by incorporating nanoscale features on that surface, with the understanding that if we reduce this resistance, we will be reducing the limiting Johnson noise. So we went ahead and fabricated these devices. We started with a high resistivity silicon wafer, oxidized on both sides using a thermal oxidation process. We then, using lithography and an etch process, we made micropillars. There was oxide on top. And then we oxidized this surface, and that gives us a selective oxidation, which, when we remove the oxide, yields silicon tips that are very sharp at the tip. This is an established technique, although it's difficult to understand why this happens, but there is selectivity, and the oxide grows faster towards the tip as compared to the side of that pillar. So you end up with these sharp tips. Now, these sharp tips, that surface, we remove the silicon dioxide there. We then grow LPCVD nitride on the surface here, which is shown in red. We then use backside alignment and traditional bulk wet etch techniques to remove the silicon from the backside. And that basically leaves us with a membrane of silicon nitride in the shape of these tips. So this is a mold which we fill with metallization from the backside. In this case, it was a layer of gold. We then isolate these two electrodes. And the reason we do that is because our measurements are done in a differential mode. So we provide 2 electrodes per die that we make. So this isolation gives us 2 electrodes. Remember, this is high resistivity silicon. So we get a good isolation between these 2 electrodes. We then bonded wires on the back side and then covered everything on the back with this purple insulating elastomer to seal the back side. These are some pictures from fabrication. These are the sharp tips that we make in silicon that form the basis of the mold. These are about 2 micrometers high, about 10 nanometers in tip. And we can control this thickness of the tip, as you'll see downstream. This is a computer-generated image showing, it's simplified, showing what our electrode looks like on the business end, really. So we have 1, 2, 3, 4, 5, 6 clusters of arrays of these gold tips. Each one here is shown for simplicity to be a 5 by 5 array. In reality, it's a 270 by 270 array. So we have about 73,000 in one of these. This is an SEM graph. These are gold tips closer up, and you can see that they are broader than the 10 nanometers, and there's a reason for that. But we can control this tip thickness. Device on the front after dicing, on the back, bulk micromachined silicon, gold everywhere, and then silver epoxy paste used to connect wires. And here's a package sensor ready for use. We then went ahead and implanted these sensors. This was done by my doctor friends at the Indiana University School of Medicine. And on your left here, you see a picture of an exposed stellate ganglion. This inset in the figure on the right is a simplified version of what we wanted. We wanted to put our sensor, which consisted of 2 electrodes, and then place what they have been using conventionally, which are wire electrodes, so we could get the input from both of them and then compare the performance of our sensor. It's much more messy in the real world, so you can't see much, but this is what it is. This is our nanoelectrode. This is our planar nanoelectrode sensor. These are conventional wire electrodes. That there in the black outline is an exposed stellate ganglion. The stellate ganglion is a nerve that goes to the heart, that part there. And we then connected both these outputs from these 2 sensors to preamplifiers and to a transmitter, which is a standard off-the-shelf kind of a transmitter. This was placed outside the rib cage of the animal, and the animal was allowed to heal over a period of a few weeks, and then we measured sympathetic nervous activity from it wirelessly. Now this is a sample screenshot of data we recorded. We have time on the horizontal axis here, and then we have vertical axis, we have voltage amplitudes. We have 2 outputs. This is the conventional wire electrode. PNA is a planar nanoelectrode array. This is our sensor, and you can see qualitatively that our so this is useful information, by the way. I must clarify. This is activity, sympathetic nervous activity, and you can see that qualitatively our sensor is much more superior to the wire electrode. By the way, both of them were placed, both the wire sensors as well as the PNA were placed very close to each other on a nerve, so they were making simultaneously the same measurements. And qualitatively, it's very evident that we are much superior. Also notice that these spikes, these are not useful information. This is EKG, so EKG noise is being picked up by the wire electrode. We're pretty clean here. Measured SNR of the sympathetic nerve activity 1 month after implantation. So 11 different data points. Just for comparison, our electrode had a footprint of 0.44 millimeters squared. The wire electrode was much larger, about 8 millimeters squared. The average signal, though, we are much stronger, 980 microvolts compared to 145 microvolts. Noise, we are lower, 4.29 microvolts compared to 36.5. Resultingly, our signal-to-noise ratio is much better than what a wire electrode would offer you, about 47 to 11.5. So the average improvement over these 11 events is about 35.5 decibels, which is quite good. And after 3 months, same trend. You see, these numbers stay the same, of course, but average signal has dropped a bit, but it's dropped both sides. And that's understandable because electrodes get poisoned with time. The RMS noise, we are still better, lower noise compared to the wire electrodes. Signal-to-noise ratio has dropped, but we still have an advantage of about 25 decibels over conventional wire electrodes. So summary and future work, this is an effort to improve the understanding of the sympathetic tone. And the first step in that direction is to improve the signal-to-noise ratio of the measured sympathetic nervous activity. We fabricated electrodes with increased surface area, implanted them in canine test subjects, successfully measured sympathetic nervous activity with improved signal-to-noise ratio over traditional wire electrodes for up to 3 months after implantation. The electrodes don't die after that. It's just that the transmitter has a limited battery life, so we had to take it back out of the animal. The animal is still alive, and so experiments had to be terminated at that end. In future, we want to continue measurements to establish a database between different stimuli and what kind of SNA we measure as a result, and try to establish a biophysical relation between sympathetic nerve activity and life-threatening diseases. And of course, electrical and mechanical characterizations of the PNAs. There are a lot of unknowns at this point in time. We need to answer a lot of questions. So a lot of work needs to go in this direction. I would like to thank my colleagues at the IU School of Medicine who've worked very hard to make this possible. And of course, my friends and colleagues at Purdue University who've helped me all along. Thank you very much.